Within this leopard are trillions of cells, each with the same exact genetic code. The cells in his eyes, the cells in his nose, and in his ears, all the same DNA. And yet, each cell plays a different role in keeping this cub alive. While all of his cells possess the same genes, not all genes are expressed in all cells. And this is what gives each cell its different function. The eye will express different genes and make different proteins than the nose, so that the eyes can see prey and the nose can smell it. Today, we're going to explore prokaryotic gene expression, specifically operons. I'm working on a schematic and a few materials that would be perfect for your notes. As always, links will be below. Our lesson agenda will include prokaryotes versus eukaryotes, some basics on gene expression, including transcription regulation, and then we'll move into operons, two types, repressible and inducible, and different types of regulators, specifically negative and positive, and we'll be using the lac operons and Tryptophan as examples. And this chapter had a lot of requests, so without further ado, let's go. DNA is, of course, the written genetic code. This molecule contains directions for all RNAs and proteins that can be potentially expressed within the cell. It also contains the code for the proteins that regulate which of those genes can and will be expressed. Gene expression is the process in which information from a gene is used in the synthesis of a functional gene product. Typically, this is a protein, but science is revealing more roles of different types of RNA in the cell. Gene expression is what determines whether the leopard cells will become eye cells or nose cells. Those gene products that we discussed are RNA and protein. This should look familiar. It's a concept in biology known as central dogma, which states that information flows from DNA to RNA to protein. The first stage in red, where DNA base pairs are read to form RNA, is called transcription. The second stage in yellow, where the RNA codons are read to make protein, is called translation. Organisms can regulate gene expression at various stages of this process. We need to distinguish between prokaryotic and eukaryotic gene expression regulation because they're very different. This lecture will be broken into 3 videos. In this video, we're focusing on prokaryotic gene expression regulation. In part 2, we'll cover eukaryotic gene expression regulation, including chromatin transcription and post-transcriptional modifications. And in part 3, we'll dive into gene expression in embryonic development and cancer. Quick review of eukaryotic versus prokaryotic cells. Eukaryotic organelles are membrane-bound, and this compartmentalization of enzymes allows for specific functions. Prokaryotic organelles are not membrane-bound. Everything is free-floating in the cytosol. The big one to note for this lecture is, of course, the nucleus, which is only in eukaryotic cells. Prokaryotes have a nucleoid region, and DNA is freely floating in the cytosol. Also note that while you and I have 46 linear chromosomes, prokaryotes typically only have 1 circular chromosome in the nucleoid region. Because of this, transcription and translation can occur simultaneously in prokaryotes. Okay, back to the topic at hand, gene expression. It's important to keep the key differences between prokaryotic and eukaryotic cells in mind because they regulate their gene expression very differently. Prokaryotic transcription and translation occur simultaneously in the cytoplasm. Why does this make sense? Well, no organelles means everything is floating around in the cytoplasm together, so it all has access to the DNA at the same time. Gene expression regulation occurs primarily at the transcriptional level here. Eukaryotic gene expression is regulated during transcription and RNA processing, which take place in the nucleus, and during protein translation, which takes place in the cytoplasm. Further regulation may occur through post-translational modifications of proteins in both prokaryotes and eukaryotes. And don't worry, we're going to hear these terms over and over in this lecture, and we'll break down what each mean. And in case you didn't catch everything, here's a table that summarizes the differences we've emphasized between eukaryotic and prokaryotic gene expression so far. For the rest of this lecture, we'll focus on prokaryotic gene expression regulation. This will cover operons and their components, negative versus positive gene regulation, including the two types of negative gene regulation, repressible and inducible. Let's get back to this little guy. Remember, all of his cells contain the same DNA, the same genes are in his skin, heart, and eye cells, but every cell does not express all of its genes all of the time. The big question here is, how can two cells with the same set of genes function differently? And here's the answer. To be expressed, each gene requires a particular set of transcription factors. Different cells have different sets of specific transcription factors. Due to this, every cell does not express all of its genes all of the time. We're going to return to transcription factors in just a minute, so put that term in your back pocket. Cells control when a gene is expressed to make RNA and protein, how much of the protein is made, and when it is time to stop making that protein because it's no longer needed. The control of gene expression is extremely complex. Like the cells of our leopard friend here, it would not be good if the cells of the eye expressed the proteins involved in growing fur. And in case I haven't mentioned yet, this process is extremely complex. Malfunctions in this process are detrimental to the cell and can lead to the development of many diseases, including cancer. In part 3, we'll go into detail about this. Okay, so we're all on board that gene expression is highly complex and controlled by the cell. When a gene is expressed, the products of RNA and protein are made. Of course, enzymes are a major category of proteins expressed by the cell, as they are the ones that build and break bonds. Here's a quick recap of enzymes. If you need more detail, I have an entire lecture dedicated to enzymes and metabolism called Chapter 8, Part 2. Enzymes are catalytic proteins that speed up metabolic reactions. They do this by lowering the activation energy of a reaction. On the left is a 3D rendering of an enzyme, which is a chain of amino acids folded into the perfect conformation to allow it to bind to its substrates. For example, the enzyme sucrase breaks down the sugar sucrose into glucose and fructose. Remember, enzymes are very specific for the reaction that they catalyze. Now, a cell can regulate the production of enzymes in two ways: feedback inhibition and gene regulation. I discussed feedback inhibition in detail in my Chapter 8 lecture as well. And just so you know, I have materials available in my Send Owl store. In feedback inhibition, the end product of a metabolic pathway shuts down the pathway. In the example of enzymes, the end product shuts down further synthesis of the product by inhibiting enzyme activity. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. Let's look at this example. Again, in feedback inhibition, the end product of a metabolic pathway shuts down the pathway. Here, enzyme 1 is binding to a substrate and creates intermediate A, which then binds to enzyme 2, creating intermediate B. In the last step, intermediate B binds to enzyme 3, creating an end product, the yellow square. The yellow square indicates that this pathway was successful in creating what the cell needed. Over time, a large amount of end product, the yellow square, is created, and it will naturally disperse through the cell and find enzyme 1. It will bind enzyme 1 and inhibit it to shut down the pathway. Again, this conserves resources and energy for the cell. We don't want more yellow square product than's needed. This is feedback inhibition. The second method of enzyme regulation is gene regulation, which is the focus for us today. Cells can adjust the production level of certain enzymes by regulating the expression of the genes that code for those enzymes. The control of enzyme production is thus at the level of transcription. Here it is again, transcription. We'll get to this in a moment. One basic mechanism for this type of regulation, where we can control the expression of groups of genes, is called the operon model. To simplify this, we can regulate enzymes at the level of activity, And we can regulate enzymes at the level of production. Make sure you see and understand this difference. Feedback inhibition is not changing the amount of the enzyme, it's only changing the activity of the enzyme. The pathway's product is inhibiting the enzyme. Gene regulation is changing the amount of the enzyme. The cell can turn on and turn off the expression of that enzyme. And this is very complex, and this is what we're discussing in this lecture. We'll be learning about the operon model, which is how prokaryotes regulate gene expression at the level of transcription. So we're back to transcription factors. Remember, to be expressed, each gene requires a particular set of transcription factors, and different cells have different sets of specific transcription factors. We learned about transcription factors in my chapter 11 video called Cell Communication. A transcription factor is a protein that controls the rate of transcription of a gene by binding to a specific DNA sequence and altering RNA polymerase recruitment. They are sequence specific, meaning they're looking for a particular place on the DNA to bind. Now, look at this image. We have the DNA double helix. It's uncoiled so that the transcription machinery can bind to it. Directly bound to the DNA is the transcription factor, and interacting with that is the RNA polymerase. Recall that RNA polymerase is the enzyme that reads the DNA to build the RNA transcript. Transcription factors can work alone or with other proteins in a complex. Their goal is to either activate or repress recruitment of RNA polymerase to specific genes. I'm going to repeat that. Their goal is to either activate or repress recruitment. Recruitment of who? RNA polymerase to specific genes. The area of the transcription factor that binds directly to the DNA has a special name. It's called the DNA binding domain, DBD. And where can they bind to DNA? They bind to enhancer or promoter regions of DNA. Prokaryotes, like bacteria, often respond to environmental change by regulating transcription. Their DNA is organized into these circular chromosomes, which you can see in the bottom left. And this DNA sits supercoiled in the nucleoid region of the cell. Remember, prokaryotic transcription and translation occur simultaneously in the cytoplasm. When the resulting protein is no longer needed, transcription stops. When more protein is required, more transcription occurs. Therefore, in prokaryotic cells, the control of gene expression is mostly at the transcriptional level. Proteins that are needed for a specific function or that are involved in the same biochemical pathway are encoded together in blocks called operons. For example, all of the genes needed to use lactose as an energy source are coded next to each other in the lactose or lac operon and transcribed into a single one mRNA. Even though there are multiple genes, we make one single mRNA. The operon will be the focus of discussion for the rest of this video lecture. Take a moment to familiarize yourself with the basic structure of an operon. Notice there is usually a regulatory gene upstream. Upstream is the terminology used to describe a location on a gene. It means to the left, and it's called upstream because it comes first in the flow of information. Everything else is downstream of this. Then there's usually a span of non-coding DNA, followed by the promoter and operator, and finally the structural genes. We'll go into detail about these very shortly. The structural genes are transcribed into mRNA. Notice they are all on one strand of mRNA, even though there are multiple genes. This is the beauty of the operon. Finally, the mRNA is read by ribosomes and translated into protein products. This is central dogma. The flow of information is DNA goes to mRNA goes to protein. The operon is simply defined as a cluster of genes transcribed into a single mRNA molecule. The beauty of operons is that the cluster of functionally related genes can be coordinately controlled by a single on-off switch. The switch is a segment of DNA called an operator, positioned within the promoter or sometimes between the promoter and genes. And as we saw in the previous slide, an operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. Let's get into the components now. The promoter. Recall from chapter 17 on DNA replication that the promoter is the sequence of DNA to which proteins bind to initiate transcription. RNA polymerase, shown here, is the enzyme that reads the DNA and builds the mRNA. It needs to be able to bind to the DNA. Promoter DNA sequences provide a secure initial binding site for RNA polymerase and for proteins called transcription factors, which recruit RNA polymerase. Think of the promoter like you'd think of the promoter of a big event. The promoter's job is to recruit the appropriate people so that this event can happen. It's a big deal. You can't have an event without a promoter. For the cell, the big event is gene expression. Instead of people, the DNA promoter is recruiting the correct transcription factors and DNA polymerase so we can get this gene party started. Now we talk about the operon having an on-off switch. This is called the operator. The switch is a segment of DNA called an operator, positioned within the promoter or sometimes between the promoter and the enzyme coding genes. The operon can be switched off by a protein repressor, and we'll see this in a moment. To help remember the role of the operator as the switch, think of the switchboard operators of the early 20th century. You may have seen something like this in a movie or show. A caller picks up their phone and is connected with usually a female operator. The operator uses their switchboard to connect them to their desired call receiver. Just look at that switchboard. And at any moment in time, the operator can switch the call on or off, just like our operon operator, which can switch the operon on or off with the help of the next component. The repressor. The operon can be switched off by a protein repressor binding to the operator. This literally creates a physical blockade. The repressor prevents gene transcription by binding to the operator and blocking RNA polymerase from traveling along the DNA to read it. Now, where does this repressor come from? The repressor is the product of a separate regulatory gene located some distance from the operon itself. It's shown here upstream. Now, this is where it gets a little tricky. The repressor can be in an active or inactive form, depending on the presence of other molecules. A co-repressor is a molecule that works with a repressor protein to switch an operon off. It does this by binding to the repressor. For example, E. coli can synthesize the amino acid tryptophan when it has insufficient tryptophan. Tryptophan is a co-repressor, as we will see shortly. The tryptophan operon will be the first one that we focus on in this lecture. By default, the tryp operon is on, and the genes for tryptophan synthesis are transcribed. To help you remember this, think of a consequence of a person tripping near a hill.
Oh, man!
Anything they have will continue down the hill until something stops it, until something represses it. Same here. It is on by default, meaning the genes are being expressed. Expression will only stop with a repressor. So if the default for this operon is on, how do we stop gene expression of the tryp operon? The answer is tryptophan itself. This is a type of negative feedback. When tryptophan is present, it binds to the tryp repressor protein, which turns the operator off. Notice that tryp is a co-repressor. And by the way, where does the repressor protein come from? It's encoded in the regulatory gene TRPR, TRPR. Again, seen upstream of the promoter. TRPR is transcribed into mRNA and translated into the inactive repressor protein. The repressor begins in an inactive state. The repressor is only in the active state in the presence of its co-repressor tryptophan. This allows it to bind to the operator to block transcription. Thus, the tryp operon is turned off or repressed if tryptophan levels are high. And just think why this makes sense for a moment. The purpose of this operon is to make enzymes that synthesize tryptophan. If there is already high levels of tryptophan, the cell doesn't need to make more. So the tryptophan itself can act as a signal to the cell to halt expression of the tryp operon and thus stop making more tryptophan. When cellular levels of tryptophan drop, operon gene expression will increase. If this is a little confusing, don't worry, we're going to walk through this. When tryptophan's present, tryptophan will act as a co-repressor and bind to the repressor protein. This causes the repressor to become an active repressor. This active repressor binds to the operator in the promoter region of the TRP operon, which blocks transcription. As tryptophan accumulates, it inhibits its own production by activating the repressor protein. To make this easy, highlight, star, and underline this. Whatever you have to do to bring attention to this sentence. Tryptophan present, repressor active, operator off. In the absence of tryptophan, the cell needs to make some, so we're going to turn the operator on. Because there is low tryptophan, the repressor does not have its co-repressor, so it is inactive. RNA polymerase attaches to the DNA at the operon's promoter and transcribes the operon's genes. Highlight this sentence here. Tryptophan absent, repressor inactive, operator on. Here's a comparison of the two states with and without tryptophan for your notes. Again, notice that when tryptophan's present, it's binding to its repressor, and the repressor is going to bind to the operator and block RNA polymerase, and this turns the tryp operon off. When tryptophan is not present, the repressor is not activated, it's not bound to the operator, and RNA polymerase can read the operon. So let's continue with the operon on. We want to express the genes that make tryptophan. The tryp operon is a region of the DNA that includes from left to right the promoter, which includes the RNA polymerase binding site and the operator, followed by operon genes tryp E, tryp D, tryp C, tryp B, and tryp A. As the RNA polymerase works from left to right, a single, long, continuous strand of mRNA is transcribed from the operon genes with start and stop codons for each gene. Check out this image. Notice the single strand of mRNA, 5' to 3', with start codons in green and stop codons in red. In between each start and stop codon are the RNA codons that will be translated into a gene. So how many genes does this mRNA code for? It codes for 5. Following transcription, operon genes are translated by ribosomes into polypeptide subunits that make up enzymes for tryptophan synthesis. Those polypeptide subunits are E from tryp E, D from tryp D, C from tryp C, B from tryp B, and A from tryp A. And once enough is made, tryptophan will act again as a co-repressor by binding to the repressor protein, and this will repress the operon and turn it off. Now you understand how the tryp operon works. Knowing what you know now, what kind of operon would you categorize this as? Repressible or inducible? Tryp is a repressible operon. A repressible operon is one that is usually on. Binding of a repressor to the operator shuts it off. We saw that when tryptophan, the co-repressor, binds to the repressor, the operon is shut off. We're about to learn a few other kinds of operons. Repressible operons are just one type of negative gene regulation. And if you guessed that there's also positive gene regulation, you're correct, but we'll get to that later. For now, negative gene regulation. There are two types, repressible and inducible. An inducible operon is one that is usually off. A molecule called an inducer inactivates the repressor and turns on transcription. So it's basically the opposite of what we just saw. The example we will learn is the lac operon. Notice here that both kinds of negative gene regulation have a repressor. The difference lies in the molecule that binds to it. We either have a co-repressor or an inducer, and they have opposite effects. Regulation of the tryp and lac operons involves negative control of genes because operons are switched off by the active form of the repressor. Repressible enzymes usually function in anabolic pathways, pathways where we are building something. Their synthesis is repressed by high levels of that end product, as tryptophan was. Again, this is a type of negative feedback. Inducible enzymes usually function in catabolic pathways, pathways where we are breaking something down. Their synthesis is induced by a chemical signal. Here's a table illustrating the differences between catabolic enzymes and pathways and anabolic enzymes and pathways, which here are written as biosynthetic. This is a really good resource to help you organize all of this information. Again, catabolic pathways catalyze breakdown of nutrients to generate energy, or more precisely ATP, the energy currency of the cell. In the absence of the substrate, there is no reason for the catabolic enzymes to be present, and the operon encoding them is repressed. In the presence of the substrate, when the enzymes are needed, the operon's induced or de-repressed and turned on. Let's look at the lac operon as an example of an inducible operon. By default, the lac operon is off and must be induced to turn it on. To help you remember this, think of the word lack, but L-A-C-K. When you lack something, you don't have it. Here we lack gene expression of the lac operon. To turn it on, it needs to be induced. So if the default for this operon is off, how do we induce gene expression of the lac operon? The answer is an inducer. And in this case, the inducer is lactose. Kind of. It's actually allolactose, which is a very similar structure to lactose. It's an isomer of lactose. This operon is called the lactose operon because its job is to code for enzymes used in the hydrolysis and metabolism of lactose. In other words, these enzymes break down lactose after it's been ingested or taken in by a bacteria so that the bacteria can use it as energy. Just like the trip operon, entire transcription unit is under the control of one main operator and promoter and a regulatory gene. Here it's LAC-I, located outside the operon, upstream from the promoter, encoding the repressor protein that can switch off the operon. So we can see the repressor sitting on the operator, which keeps the operon off. This is the default for this operon. So how do we turn it on? When allolactose is present, it signals to the cell that lactose is also present. In fact, again, allolactose is just an isomer of lactose. Remember, the job of the lactose operon is to make enzymes that break down lactose. By itself, the lac repressor is active and switches the lac operon off. Now watch the repressor. Allolactose binds to the active lac repressor protein, altering the shape of the repressor so it can no longer bind to the operator sequence on the DNA. And voila, the operon's turned on. But I do want to note right now that this is only allowing slow rates of expression, which is why I have a little snail here. And we're going to learn why that is later. Here's a summary of what we've covered so far with the lac operon. When lactose is absent, the repressor is active and the operon is off. When lactose is present, repressor is inactive and the operon is on. So here's a schematic of the lac operon so far. When lactose and allolactose are present, the operon's turned on, RNA polymerase can bind to the promoter region, and transcription of the lac operon occurs from left to right. A continuous strand of mRNA is transcribed with start and stop codons for each of the operon genes. The results of translation for each gene transcribed from the operon are LAC-Z, which is beta-galactosidase, LAC-Y, which is permease, and LAC-A, which is transacetylase. These enzymes are all involved in the hydrolysis and metabolism of lactose, again, also that we can use lactose, break it down for energy. However, what we've seen is the negative regulation of the lac operon. There's also a positive regulation mechanism. In positive gene regulation, an activator protein stimulates transcription by binding to DNA, and this is usually not at the operator. A stimulatory protein that we will see in this lecture is CRP, which is cyclic AMP receptor protein. You should remember cyclic AMP or CAMP, CAMP, from the cell communication chapter. It's often a secondary messenger in the cell. The actual structure is pictured on the far right, but for this lesson, we'll use a yellow circle to show CAMP. The stimulatory protein here is the blue CRP. When CAMP is present, it binds to CRP like so, and can then act as a transcription factor, and we'll see this shortly. I also want to note that CRP is also called CAP in some textbooks. Remember, we're talking about prokaryotes here, and specifically E. coli. E. coli prefers glucose as an energy source, just like we do. Only when lactose is present and glucose is in short supply or not at all does E. coli use lactose as an energy source. Only then does the lac operon get turned on. And let's see how this happens. First, where does CAMP come into this? When glucose levels drop, there's a rise in cyclic AMP and it accumulates in the cell. And this is because CAMP is involved in glucose metabolism. And because there's no glucose, it doesn't get used up in that pathway and it rises and it accumulates. The accumulating CAMP molecules will bind to the CRP proteins. The complex then binds to the promoter region of the genes that are needed to use lactose. Here's a complete schematic of the lac operon turned on and de-repressed. Lactose is present, so the repressor is inactive and removed from the operator. The CRP camp complex is bound to the promoter, and this lets us know that there's low glucose because there's high CAMP. And the RNA polymerase is also bound to the promoter. This operon is ready to quickly express genes for the metabolism of lactose. In these operons, a CRP binding site is located upstream of the RNA polymerase binding site in the promoter. Notice how I have two colors here to distinguish the two binding sites. They're still both part of the promoter. When CRP binds, it stabilizes the binding of RNA polymerase to the promoter region and increases the affinity of RNA polymerase, thus accelerating transcription. And now I have a guy running really quickly to show fast transcription. Remember, before I had a snail for slow transcription. So let's look at that one more time. The lac operon by default is off and must be induced. This will only happen when glucose is low and lactose is present. When glucose is scarce, CRP, also called CAP, is activated by binding with CAMP. Activated CRP attaches to the promoter of the lac operon and increases the affinity of RNA polymerase, thus accelerating transcription. The lac operon is subject to positive control through a stimulatory protein that activates transcription. When glucose levels increase, CRP detaches from the lac operon and transcription returns to normal. Low-level CRP helps regulate other operons that encode enzymes used in catabolic pathways. Remember, catabolic pathways usually involve inducible operons like the lac operon. The ability to catalyze compounds like lactose enables cells deprived of glucose to survive. However, notice what is happening in this final example. The CRP-CAMP complex is bound, but so is the repressor. So what is this telling us about what energy sources are present? We know there's no glucose because CAMP is present, but what about lactose? The repressor is still bound. This tells us that there is no lactose because there's no allolactose. Both things must be true, low glucose and lactose present, for the lac operon to be turned on. And this should make sense. Why would the cell want to waste energy coding for genes it can't use? So remember, the compounds present in any given cell determine which genes are turned on. So that's it. We've covered the regulation of the lac operon. With all of that information, it's crucial to see things together to draw comparisons and to contrast. Here's another way of looking at what we've just learned. There were two control mechanisms. One used lactose and the other used glucose. The first control mechanism is negative control. The second mechanism is positive control. And there were different regulator proteins for each case. Here's a comparison chart of negative and positive regulators of the lac operon. Here, our negative regulator is the repressor protein. When active, it attaches to the operator and prevents RNA polymerase from binding. The presence of allolactose, the isomer of lactose, acts as an inducer. It inactivates the repressor, which then dissociates from the DNA. The positive regulator is the CRP-CAMP complex. When it is attached to the upstream region of the promoter, it supports RNA polymerase binding and results in fast transcription. Both of these have to happen simultaneously for expression of the Lac operon. Here's a table of control elements of the Lac operon for notes. This includes both negative and positive gene regulation and the outcome. Notice that if no lactose is present, this operon will not be expressed no matter the glucose levels. This makes sense. We don't want to turn the Lac operon on if there is no lactose. The other possibilities are related to glucose. If glucose and lactose are present, the Lac operon will be on at low transcription rates. If glucose is not present and lactose is present, the Lac operon will be on at high, fast transcription rates. Here's one more example of the Lac operon control elements. I won't go through this again, but if you download my notes, this will be included. And well done for getting through a pretty difficult topic in biology. Thanks for watching. Keep an eye out for part 2, where we're going to go into eukaryotic gene regulation. Best of luck, and I'll see you next lecture.